I'm Jonathan Richard from Andres Finzi Lab, and today I'm really happy to present our latest work in collaboration with Preeti Kumar, in which we modulated HIV-1 envelope conformation to decrease the HIV-1 reservoir. There is increasing evidence suggesting that the conformation adopted by the HIV-1 glycoproteins at the surface of infected cell substantially impact antibody recognition. So in its unbound form, the envelope adopts a close conformation that is mainly resistant to ADCC-mediating non-neutralizing antibody naturally present in the plasma from infected individual. So these antibody target epitope that become only exposed when the envelope interact with CD4, the cell surface, thus adopting a more open CD4-bound conformation. However, the virus evolved several strategies to prevent envelope and CD4 interaction at the surface of infected cell. Notably, the viral protein F in VPU downregulates cell surface CD4. So this keeps the envelope in its close conformation, and consequently, infected cells were found to be mainly resistant to recognition and ADCC mediated by HIV-1 positive plasma. So to bypass this protection, we demonstrated that we can use small CD4 mimetic compound to force the HIV-1 envelope to adopt the CD4-bound conformation despite NEF in VPU expression. And as you can see in blue, this significantly enhanced the capacity of HIV-1 plasma to recognize infected cell and to sensitize these cells to ADCC responses. This sensitization of infected cell to ADCC requires sequential opening of the envelope trimer and involves two families of non-neutralizing antibody present in the plasma from infected individual. So we demonstrated that the CD4 mimetic compound sufficiently opened the envelope trimer to expose the co-receptor binding site. Then the bivalent interaction of co-receptor binding site specific antibody further opened the envelope trimer to allow binding of anti-cluster A family of antibody. Then the Fc portion of both classes of antibody are required to efficiently recruit Fc gamma receptor bearing effector cells such as NK cell to mediate the elimination of HIV-1 infected cell by ADCC. Antiretroviral therapy efficiently suppresses HIV-1 replication but does not eradicate the virus. So virus reactivation and rebound from latent reservoir occur in most individuals after treatment interruption. So in the study, we decided to evaluate the therapeutic potential of CD4 mimetic. So in collaboration with Preeti Kumar at Yale University, we use SRG15 humanized mice that support efficient development of functional NK cell and antibody effector function to evaluate the potential of using CD4 mimetic compound in combination with these non-neutralizing antibody to reduce the size of the reservoir in vivo and to delay viral rebound after treatment interruption. To evaluate the impact of such treatment during acute infection, we generated SRG15 humanized mice using human PBL. Humanized mice were then infected with HIV-1 GRCSF. After confirming plasma viremia at day 7, mice were treated with CD4 mimetic in combination with control plasma, HIV-1 positive plasma, control IgG, or co-receptor and cluster A antibody. We observed a significant reduction in the plasma viral load in all mice that were treated with the CD4 mimetic, which is consistent with the inhibitory activity of the compound against HIV-1. This was associated with the restoration of peripheral CD4 T cell. To measure HIV-1 reservoir, we evaluated the level of CD4 T cell associated HIV-1 DNA in multiple tissue. And as you can see here, we observe a significant decrease in the HIV-1 reservoir only in the mice that were treated with the combination of CD4 mimetic with either HIV-1 positive plasma here in blue or with co-receptor binding site and cluster A antibody here in pink, suggesting that such treatments significantly reduce the size of HIV-1 reservoir in vivo. We observed no reduction in the bone marrow, which was consistent with the poor penetration of the CD4 mimetic in this tissue. To evaluate the role of Fc effector function, we introduced LALA mutation in the Fc portion of both classes of non-neutralizing antibody. These mutations were shown to decrease antibody interaction with Fc gamma receptor. So compared to wild-type antibody that mediates strong ADCC response against infected primary CD4 T cell in the presence of CD4 mimetic, introduction of these mutations in co-receptor binding site or cluster A antibody totally abrogated this ADCC response. In vivo, treatment of infected humanized mice with the combination of CD4 mimetic and wild-type antibody again significantly reduced the size of the reservoir in multiple tissue, and introduction of the LALA mutation significantly reduced this activity, confirming the role of Fc effector function in the reduction of HIV-1 reservoir mediated by CD4 mimetic compound. To evaluate the role of NK cell in this phenotype, we generated SRG15 humanized mice using human PBL depleted or not of NK cell. So in infected humanized mice expressing NK cell, again we observe a significant decrease in HIV-1 reservoir in multiple tissue upon treatment with the combination of CD4 mimetic with either HIV-1 positive plasma or co-receptor binding site and cluster A antibody. In contrast, little to no change was observed in humanized mice devoid of NK cell, confirming the key role of the NK cell in this reduction of HIV-1 reservoir. To study HIV-1 reservoir after heart interruption, we infected SRG15 humanized mice generated with hematopoietic stem cell. Mice with established infection received daily heart regimen to suppress HIV-1 viremia for at least 1 month. After heart interruption, mice were treated with CD4 mimetic and/or human plasma or human IgG. As you can see here, all mice in the control cohort receiving CD4 mimetic or control antibody or plasma alone or in combination show no significant difference in time to viral rebound compared to the mock-treated mice. In contrast, we observe a significant delay in the viral rebound only in mice that received the combination of CD4 mimetic with HIV-1 positive plasma or co-receptor binding site and cluster A antibody, demonstrating that these approaches significantly impact the size of competent HIV-1 reservoir in vivo. Importantly, no escape mutant were identified among the rebounding virus. To evaluate the therapeutic potential of CD4 mimetic in heart-treated patients, we isolated CD4 T cell from 2 heart patients and upon activation were able to reactivate the HIV-1 replication ex vivo, as you can see here. And we found that the capacity of autologous plasma to recognize HIV-1 infected cell and to mediate ADCC response was strongly enhanced in the presence of CD4 mimetic. To recapitulate this result in vivo, we generated SRG15 humanized mice using PBMC from these 2 donors. Syndromenic activation of transplanted T cell induced viral outgrowth from the latent reservoir in vivo, as you can see here. 3 mice per PBMC donor were either treated with CD4 mimetic alone in gray or in the combination of CD4 mimetic and autologous plasma in blue. You can see that treatment with CD4 mimetic alone significantly reduced viral load, but we detected viremia in all treated mice. In contrast, in mice treated with the combination of CD4 mimetic and autologous plasma, we detected no viremia for 5 of the 6 mice after treatment interruption, while we detected viral rebound in 2 of these mice. We detected no viremia for the 3 remaining mice, suggesting that combination of CD4 mimetic with autologous plasma is sufficient to eliminate HIV-1 infected cell and to facilitate virus control in vivo. In conclusion, we demonstrated that brief treatment with CD4 mimetic and CD4-induced antibody or HIV-1 positive plasma significantly decreased HIV-1 replication, the virus reservoir, and viral rebound after heart interruption. These effects require Fc effector functions and NK cell, highlighting the importance of ADCC. Viral rebound can be suppressed in HIV-1 positive donor cell-derived immunized mice supplemented with autologous plasma and CD4 mimetic, suggesting that CD4 mimetic could have therapeutic utility in infected individuals for decreasing the HIV-1 reservoir or achieving a functional cure. Finally, I would like to thank everybody that contributed to that work, my director Andres Fienzi, our collaborator, especially Dr. Kumar and Rashekar, who generated the data with the humanized mice. I would like to thank our founder and you for your attention.